Moving on, talking about really mostly from this perspective, but we will switch between this picture and this picture for this video. So let's talk about our nephrons in general. So nephrons, they come in two different kinds. There's what are called cortical nephrons, which are illustrated on the left side over here, and there are juxtamedullary nephrons. About 80% of your nephrons are cortical nephrons. Cortical nephrons are basically the same as juxtamedullary nephrons, but their obvious difference is anatomical and functional. The anatomical difference here is that one has a much longer loop of Henle that goes all the way down nearly to the bottom or the deepest parts of the renal pyramid, whereas the cortical nephrons are mostly in the cortex, and that's where their name comes from. Now that's the anatomical difference. What that means functionally is that if you remember from the function of the kidney and what's going on physiologically here is that your kidney is working to create a concentration gradient where the deeper regions of the renal pyramid are much more salty than the more superficial regions. So it gets saltier and saltier as you go to the bottom, basically. And we won't get into the details of how you form that concentration gradient, but the reason we do that is because that saltiness on the outside of the loop of Henle on the outside of really everything. It's the whole depth, right? All of the tissue here, with the exception of the spaces within the collecting duct and the loops of Henle, all of the area is going to be much more salty down here. And so what that allows us to do is release hormones that allow aquaporins, these little tubes that carry water, to be inserted into the collecting ducts. And when the stuff that's in the collecting duct is less salty than the stuff that's outside of the collecting duct. And because of that, if you poke a hole in the collecting duct, the water will move passively, meaning you don't have to do anything, you just poke a hole, and water will essentially flow out of the collecting duct and back into the medulla of the kidney. And that allows us to recover water. So you can sort of think of the kidney as whenever aquaporins are inserted into the collecting duct here, you are actually drinking essentially the fluid that you would have peed away. So in times when you're dehydrated or potentially too salty, basically when you need water, you sip it or remove it from the collecting duct through passive osmosis. And the only reason that is allowed to happen is if you can keep this area of the kidney salty. How do you keep that area of the kidney salty? You use juxtamedullary nephrons. And so the juxtamedullary nephrons, though they are the minority, they're only 20% of the total number, they are by far the most important for water balance. Because if these aren't running, this is the only thing keeping things salty. It's like a salt maker. It's just keeping everything nice and salty down here. If these die for any reason, the kidneys are not going to work to recover water. And so what we're going to talk about the rest of this video is mostly the blood flow through the kidney. And the reason, not through the whole kidney, really through the small microscopic structures of the kidney. The reason that topic is so important is that these are the longest capillary beds in your body, which means if you have an issue that causes blood flow to be challenging for some reason, then the kidney is what is going to be affected. And this is why renal failure and kidney disease is an extremely common thing, because many things can affect our ability to have blood move through the smallest blood vessels. And the skinniest blood vessels are capillary beds. And the longest capillary beds are what you're looking at right here. So let's talk about that blood flow. So the renal circulation begins, actually is not written in order in your list, sorry. It doesn't begin with the glomerulus. If we go down to this picture, blood is going to come in over here through the afferent arteriole. Blood will then flow through the glomerulus and out to the efferent arteriole. Efferent means to move away. Afferent means to come towards, and so those names should make some sense. You can just remember E for exit, though. It's a nice little quick way to remember it if you struggle with flipping those terms around. What is not shown very well in many of these models, but if I were to design this model, what I would add to it is the next term, the peritubular capillary bed. And where the peritubular capillary bed is, it comes, just like on the list, it comes after the efferent arteriole. So it would continue here. And it would be actually a second capillary bed, the glomerulus being the first. It would be a second capillary bed that continues and then would parallel or run along the proximal convoluted tubule over here on the right. So if we go back to this model and we zoom in here, basically afferent arterial, glomerulus, efferent arterial, and then you can see this kind of elaborately sprouting attempt to represent the paratubular capillary beds. It's basically a capillary bed that's very elaborate, meaning lots and lots of little branches that surround the proximal convoluted tubule, and it's in the cortex. Now, if I were to ask you about this on a test or something like that, paratubular capillary beds, those I would have to ask you on this model, but all those other structures, afferent arterial, efferent arterial, glomerulus, I'd ask you here because it's so much easier for you to see. And then that is 4 of our blood structures: afferent arterial, glomerulus, efferent arterial, the paratubular capillary beds. And then our last one is down in the medulla, and that's the vasa recta. And this really is the longest capillary bed in your body. And so this is also a sort of an attempt to illustrate the vasa recta, but this is our good one here because you can see the vasa recta coming down here and then returning to the venous blood flow. Now here's what's tricky about the model though, is in order to make things clear so that you can see the nephrons and see the blood flow, they've removed them from each other. In reality, this loop of Henle would be covered by this vasa recta. And if you look in your lab manual, you've got a picture where you've got the loop of Henle and it's just like crawling with the vasa recta. It's all surrounding it. So even though on this model, these two structures are separate, just realize that's not the correct anatomy. It's just so that it doesn't get in your way. They've just separated those two things. So in your mind, imagine you're taking a screenshot of this vasa recta and it's going to be placed right over, it's going to be scooted over to the right, covering this. And then most importantly, for the juxtamedullary nephrons, really what you should imagine is this thing here is really over the top of the loop of Henle. And so if it's over the top of the loop of Henle, that suggests its function. It's really going to be supplying nutrients and all the energy that the loop of Henle needs. The loop of Henle is incredibly energy demanding because it's creating a concentration gradient, which takes constant inputs of energy. So the vasa recta needs to be in constant inputs of oxygen mostly and sugars to be able to make sure that the loop of Henle works. So what happens and what's an example of when the loop of Henle breaks down? Well, if you have persistently high blood sugar for any particular reason, and that goes on for a long period of time, the red blood cells will actually change shape and they will not flow as easily through very narrow blood vessels. The narrowest blood vessels are your capillary beds. And so what will end up happening is you'll get this a couple different names for it, but one's called plasma skimming, basically, where if your blood trying to move into the vasa recta, the red blood cells will start to collide off of the walls and they will move slower than the plasma. So what will happen is that red blood cells will just be in a traffic jam while the plasma are like motorcycles moving around the traffic jam. And what that means is that if your hematocrit is 40 up here, but you're pumping more plasma and less red blood cells in here, then your hematocrit, your effective hematocrit or your oxygen carrying capacity is much less in the vasa recta. And that can kill the cells of the loop of Henle. If you kill the cells of the loop of Henle, do your kidneys completely fail? No, not necessarily, but what you can't do is concentrate your urine very well. And so one of the signs of this is a large volume of urine production, because if the loop of Henle isn't working because it's not getting enough oxygen, then you will be unable to concentrate your urine. And so that's one of the reasons you can see these weird urinalysis results. And we'll go over urinalysis next week and talk about that in a little more detail. So that's your most important blood flow to really understand around the nephron. The next two things are pretty quick. 2 anatomical structures and then 3 concepts here. And while we're on this picture, let's skip down to the concepts because they're pretty self-explanatory based on your experience in lecture. But filtration. Filtration is going on at the glomerulus. Filtration is just the fact that the glomerulus itself is a semi-permeable barrier. And what that means is that it lets some things out and it keeps some things in the blood circulation, which is the same as all capillary beds, but this one's just a little bit more elaborate. So filtration is only happening at the glomerulus, and that's pretty much the biggest thing I want you to know for the anatomy lab here, is that filtration happens at the glomerulus. Then tubular reabsorption versus secretion. I want you to know that conceptually, and I want you to know where it happens. Reabsorption is primarily happening at the proximal convoluted tubules. You're doing tons and tons of reabsorption, and what that means, the definition of reabsorption, is to remove things from the tubules and put them back into the blood. So reabsorption is sort of the opposite of filtration. And then tubular secretion is putting things into the tubule, but not doing it through filtration. Tubular secretion is happening technically both at the proximal convoluted tubule and the distal convoluted tubule, but it's usually talked about as being kind of a special feature of the distal convoluted tubule. But what that means is that the actual cells of the distal convoluted tubule, for example, would be pulling things from your blood, from the paratubular capillaries, and putting them into the lumen of the distal convoluted tubule. So it's an active process. Usually it's going to be taking a decent amount of energy, but it's not filtration. You're not just making holes or slits that things can't move through. You are actually actively pumping them. And that's the difference between secretion and filtration. As well as the location. Again, filtration happening at the glomerulus. Tubular secretion, we'll say, is mostly happening at the distal convoluted tubule. And then again, tubular reabsorption, proximal convoluted tubule. And then a couple other anatomical things here. We've got the macula densa and the juxtaglomerular cells, which make up what is called the juxtaglomerular apparatus. Juxta is basically meaning kind of near or positioned on, and glomerular means glomerulus. And so what you basically have here is a combination of structures. That's why it's called an apparatus. You've got one of these things which are basically modified smooth muscle cells, and these are called the juxtaglomerular cells. Pretty easy to see on the model. So all capillary beds, not just the glomerulus, but all capillary beds have smooth muscle leading towards them. When that smooth muscle dilates, it allows blood to move into the capillary bed. When it constricts, it stops blood from moving into the capillary bed. And these are constantly moving, and they're constantly changing, and they're automatically responding in order to basically kind of optimize what's needed by the surrounding tissues. So that's the juxtaglomerular cells. The other one is called the macula densa. So this is actually the distal convoluted tubule. That's what this tube is. And the macula densa is a structure in the wall of a specific length of the distal convoluted tubule. So it's not along the entire distal convoluted tubule, but it's along the walls on these, sort of on the same side that comes up right next to the Bowman's capsule and the glomerulus is the macula densa. So they don't have them color coded in this model. What they do show you is that look how much taller these cells are. So these taller cells, roughly making up maybe a third to almost a half of the circumference of this distal convoluted tubule. So basically all the ones on this side make up this macula densa. The macula densa, we're not going to go into the detailed physiology, but what's important to understand is that this distal convoluted tubule is connected to this tubule over here. So let me show you what I mean by that. If you go back to this picture, so let's imagine you're being filtered out at the glomerulus. You're traveling through the proximal convoluted tubule, down the loop of Henle, and back to the distal convoluted tubule. You're going to make a second pass right there. This distal convoluted tubule came from, essentially, the fluid in this distal convoluted tubule was the very fluid that was filtered out of the glomerulus. Sometimes when people learn this the first time, they think this is just some random distal convoluted tubule here. But it's not. It's actually the fluid that came out was into this proximal convoluted tubule. It would come back by the very same glomerulus. And what that means in general is that the glomerulus can automatically regulate itself. it can actually figure out whether or not it's over filtering or under secreting or over reabsorbing. It can do all sorts of fancy stuff because this is the input to the tubule. And this is basically when you're already done. And so if things get weird here, like your filtrate is too salty or not salty enough or whatever, things are wrong here, the glomerulus and the smooth muscle here can make changes that hope to fix that. And so it's pretty cool that you have this sort of check and balance to try to keep everything regulated. And if you want to learn about that, what that's called is, and you go over this a little bit in lecture, but it's called renal autoregulation. It's probably one of the more challenging concepts to think through, but it's a pretty important one. So for those of you going to nursing school, you may want to, if you have some extra time, do some Googling on renal autoregulation, because any time you spend cracking that open and trying to understand it will be worth your time. All right, that's it.